model which is used in Kennedy's disease research is fly models. So we're now moving up from cells to flies. And you can, flies are very easy to study. You can change their genetics. You can add the androgen receptor gene. They don't live very long. I think it's about a 2-week life cycle. They're relatively cheap to grow up, and you can look at them easily. They're in several different ways. And in this case, the flies with the androgen receptor with 12 glutamines or 52 glutamines were compared. Either they were fed nothing or dihydrotestosterone. And now, this is from Paul Taylor's work. They looked at the eyes after the flies were fed the dihydrotestosterone. And in the flies with the polyglutamine expanded androgen receptor, the eyes look worse. Normally the eyes are nice and even, they're sort of hexagonal cells, but here it's sort of disrupted. So that's one way to look at the models. What's happening? Are the proteins toxic to the cells? Are the fly eyes look normal? Or do they look like something's happened? Now another test that Paul Taylor's group did was to look at the fly larvae. How fast could these larvae crawl? Now, we'll see why this is relevant later. And in the fly larvae, so the flies hatch and then they become like little things that crawl, and then eventually they turn into the fly. They scratch lines in a plate, and then measure how far the worm goes in 30 seconds. The larvae goes in 30 seconds. And the one with the short glutamine track went about 10 lines, but the one with the long glutamine track, in the presence of dihydrotestosterone, could only go about 5 lines. So it went about half the distance. So again, this is a model of testing their motor ability.
How do you get a worm to crawl in the direction you want it to?
I don't know. I'll have to ask my brother. He's actually a fruit fly geneticist at Northwestern. So I'll check. Next year I'll get back to you. Okay, so another thing that we study in the lab is proteomics, so proteins, when and where they're made and how they interact with each other. And this shows a simple diagram of, you can imagine, Facebook, or maybe it's your e-chats, Paula. You know, people around the world connecting with each other, emailing each other, making friends, certain interactions. Some may stay out of the loop for a while, but then they come back in. And you can look at proteins in the cells in the same ways. So last year we published a paper about the androgen receptor protein interaction network, and we were looking at that in connection with prostate cancer. And we found some connections, that certain proteins had connections with prostate cancer. Now we've gone back and done the same thing with cells that make either the normal androgen receptor or the polyglutamine expanded receptor. And this slide is incredibly complex, so it's just like a network diagram, all the lines crossing and how the proteins interact. And this takes collaboration with the researchers who have very big computers and databases. But we did find certain differences in the proteins that interacted with either the normal androgen receptor or the faulty androgen receptor. So now I have to sort of go back a bit to the central dogma of molecular biology, what happens in the cell. So for a long time, it was thought that DNA makes RNA, makes proteins. So the DNA is like the architect who designs the master plans. Then you've got the blueprints, which is a copy of the RNA, and then it tells the cell which structures to build. So But then it was found out that, well, something might happen with the RNA. And I thought, okay, this looks pretty complicated. What would Ed do in this situation? So I thought, how to explain alternative splicing? So I thought of taking the RNA, it's like a train. So I dug out my son's train sets, and he's 23, by the way, he doesn't play with them much anymore. And so there's two trains. There's the wooden train, and then there's a smaller plastic train. So if you think of that like the messenger RNA, which is a copy of the DNA, then there's the full-length train. But when there's splicing occurs, all the introns are taken off. These are called exons, these are introns. And so these parts, which will eventually tell the cell which proteins to make, stay together. So you can make a train with 4 different cars, which will tell the cell to make a certain type of protein. But you can choose different parts of the train, always in the same order, but different parts. So this is a 3-car train, it's missing the engine. This is missing the passenger car. This you're missing the tanker. This you're missing the caboose. And this one only has the tanker and the passenger car. So from one messenger RNA, you can make many different messenger RNAs. And so that will tell the cell to make many different proteins. So this is how we get around the dilemma of they only found like 30,000 genes when they sequenced the human genome, but we can have many more proteins because the messenger RNA can be spliced. So looking back at our complex diagram, we started noticing some things, that some of these proteins were involved in alternative splicing. So looking back, Dr. Lieberman's group had published a paper that altered RNA splicing contributes to skeletal muscle disease in Kennedy, pathology in Kennedy's disease mice. So maybe it's involved, but there's very little published about it so far. So we wanted to do more research. And Milt Paliers, who's leading this effort in the lab, had worked with fruit flies in Dr. Paul Lasko's lab. Dr. Lasko is now the head of the biology department at McGill. So he got in touch with Paul Taylor, got the fruit flies in with the mutant androgen receptor, and then Milt and some students were able to work with these flies. So that's thanks to funding from the Kennedy's disease that Paul Taylor got the flies model going, and other researchers are able to share them and work with them. So this is just very preliminary work, but what Dr. Paliers was able to do was to get the androgen receptor to be made in wings. So this time, I guess he didn't want to look at so many eyes, he wanted to look at wings. They were flatter, easier to photograph. And this is the normal wing, and if you look at these, yep, I think there's a typo there. This should only read 12. So this should be 12, and this should be 112. So an androgen receptor with lots of glutamines, and only in the presence of androgen did it form these weird sort of structures. These bubbles here in the veins and a notched wing. This is just an air bubble. So something is happening, and then genetically you can add different proteins to the flies and see if that makes this sort of wing structure normal. So it's a way of testing for other proteins that could reduce the effect of a mutant androgen receptor.
Lenore, is the top the hormone and the bottom is without hormone?
Yeah.
Okay.
So that's minus hormone and plus hormone.
Okay.
If I had had time, I would have reversed them, but sharp eyes there. Okay, so now I'll move on to mouse models. In a few minutes. One of the tasks that, or tests that mice models are done on mouse models is the rotarod task, and that's sort of like log rolling for mice. So as far as I understand, Heather, correct me if I'm wrong, the mice are put on this roller, so you see all the mice, and then it starts up and it starts moving faster and faster, And then eventually the mice, or a mouse, doesn't have the strength to hang on, and so it falls off, and then the time is recorded as to how long it takes to fall off. So it tests muscle strength. Another test is the gait analysis. And so we have the mouse here, and its paws are painted nicely with blue paint. The back one's in blue and the front one's in red. And then it's sent down a track where it makes nice little footprints. So this is the mouse model from Katsuno and Sobu's group. And they showed that testosterone reduction, so lowering testosterone, helped the mice that had Kennedy's disease. And in this case, the rotor rod task is where they're hanging on the round rod. They castrated the mice, and they did quite well. But over time, the non-castrated mice, the mouse model of Kennedy's disease, they did poorer and poorer, and they fell off very quickly once the rod started to rotate. And just looking at the mice, this was a castrated mouse, whereas this was the mouse model of Kennedy's disease, so the muscles were smaller, they had atrophied. And here's the, just checking if Diane Mary's not here yet, but this is one of the earlier papers on her mouse model of 2004. Again, showing the footprint analysis, and this was for the normal mouse, the non-transgenic mouse, and this was the mouse that had the androgen receptor with 112 glutamines. You can see that the footprint pattern looks very different. They have an abnormal gait, it's wider, and shorter steps. And this is yet another mouse model, but this time the androgen receptor is expressed. It's a normal androgen receptor, but it is expressed at much higher levels in muscles. So now it's a point where we sort of challenge the view that it was the neurons only that were involved in Kennedy's disease. When this mouse model, which had too much androgen receptor in its muscles, developed symptoms like a Kennedy's disease mouse. So this is very interesting, and here we see the footprint analysis, where we have the back paws in blue and the front paws in red. And in the wild type mouse, so it was able to have 6.5 centimeters between its footprints, whereas the transgenic mouse, so that's the one with the expressing more androgen receptor, had a very short footprint, short length between its footprints, about 3 centimeters. But if those males were castrated, so they didn't have testosterone, less testosterone to bind to the androgen receptor, they recovered somewhat from having the disease.
Well,
Yes?
you take it to the fact that if they were castrated, they couldn't walk at all?
I assume they could.
Oh, okay.
Yes.
I don't know if you can speak to this at all or not, but a few years ago there was a study where Japanese did chemical castration.
Yes.
And they were not able to say any results.
It's called the Jasmine trial, and one of the things that came out was it showed in a certain set of patients who hadn't had disease for very long that it improved swallowing function. That if their testosterone levels were reduced by, as you said, chemical castration, medical castration, that function seemed to reduce. But overall, there didn't seem to be an improvement in the symptoms. Now, whether that trial whether it would work if it was started when men were younger, didn't have symptoms, I guess the same sort of theory was behind the dutasteride trial, where reducing the amount of dihydrotestosterone that bound to the androgen receptor. But mice models, as I say, they have their limitations. But it's the androgen, Kennedy's disease is ligand-dependent polyglutamine disease. So maybe there's some clues there, but we don't know quite what. Maybe it's because the different proteins interact with the receptor when it's bound to testosterone or dihydrotestosterone. So it's not the entire story. So how about men, male models? So of course there's lots of studies done on men, the EMGs, nerve conduction tiles, and Jeffrey Zajac from Australia noticed this in men, that they had a lot of hair. And the amount of hair one has depends on the activity of the androgen receptor and dihydrotestosterone. In fact, that's one thing that men take is the finasteride or dutasteride to block the conversion of testosterone to dihydrotestosterone so that they don't go bald. So there's something different about the androgen receptor in men, which with the polyglutamine expanded androgen receptor. So on this scale, the men with KD reported they had hairline between 1 and 2, whereas the male Australians had a hairline more about like number 3. And in this picture, which was taken last year at the conference, you can see that there are a number of men with a lot of hair. This is just an example of enrolling yourself in a study. And actually, I don't know, there's many more at the NIH that are ongoing, and it's really appreciated that you get involved and able to help the researchers figure out what's going on. Again, these are results from Jen Sobu. This is a 6-minute walk test in men, and this assesses a number of functions. Ability of muscles to work, ability of lungs to work, heart capacity. And each of these lines is like a single man tested at this point in time and this point in time. And in general, men with Kennedy's disease can walk less distance with time. Maybe this was added. No, I don't think everyone did that. Who could walk very far even though they had Kennedy's disease for a long time. So this is another way which physical studies are done to help us understand. So now this makes a connection. You can study the flies who can't crawl as far. You can study the mice who don't walk as far. You can study the men who have problems or can walk a less of a distance at a time. So all these sort of different aspects can work together. Okay, is this the same in one model? Is it not the same in one model? You can look at the cells to try treatments. You can look at the mice. You can look at the flies. Even have trials with men to see if certain compounds, certain molecules, certain proteins might help Kennedy's disease. So I just wanted to introduce you to some of the people in the lab. Some of you may remember Shazza from last year. She couldn't make it this year, but she says hi to you all. Carlos is here this year. I'm here. Dr. Tafiro is a very busy physician, chief of endocrinology at the Jewish General Hospital. Milt is the researcher who has spearheaded the fruit fly research. His wife Teresa also works in the lab. A lot of the work with the fruit flies was done by Alexandra, a summer student, and now we have a talented undergraduate student, Connor, who's working with the fruit flies. And on this side, Carlos is working with the proteasome model, as is Shazza, and Sean is working with the alternative splicing, looking at that in cells with normal and the polyglutamine expanded androgen receptor. So this is the plea. There's a t-shirt table under there for Canadians if you want a tax receipt. You can go to the Jewish General Hospital site, and they have a foundation which they have specifically set up, the Kennedy's Disease Research Fund there, until Andrew gets the Canadian branch going. And finally, I would like to thank all of you for inviting me to be here. And it's great to meet you, and I hope if you have any questions, feel free to ask them. Thank you.
Thank you.
Yes.
Referring back to San Diego's debate over the aggregate, whether it was the cause or effect, has anything been That's still up for grabs.
That's still up for grabs. Yeah.
Wow. Okay. It's a complicated subject.
Yeah.
Okay. Anybody got any questions? Okay.
Thank you.
Thank
Thank
you.
you.
I wonder what the fridge was.